Okay, so a quick review from the previous class, and also because my mic got disconnected and I didn't even notice, so there was no audio. So the chapter we covered was 1A, Chemistry for Biologists, and it covers the very basic concept of ionic and covalent bonding. Ionic bonding is the transfer of electrons between the atoms of elements and usually occurs between metals and non-metals. We give the example of sodium and chlorine, and how sodium transfers its outermost electron to chlorine, achieving a noble gas structure, and how chlorine accepts that one electron, achieving a noble gas structure as well. And what's holding, so the atoms develop charges. When you're losing an electron, that's a positive charge. When you're gaining an electron, that's a negative charge. And it is the electrostatic force of attraction between the ions, in this case now, that hold the molecule together. There are a number of important anions. Those are the ones with the negative charge that are involved in our body in biology, and similarly a number of important cations, and we'll come across these throughout the syllabus. For the anions, we've got nitrate, phosphate, chloride, and hydrogen carbonate. As for the cations, we have sodium, calcium, hydrogen, and magnesium. Next, we have covalent bonding, which is the sharing of electrons between atoms. Right, and so you can see an example of hydrogen chloride, where hydrogen needs only one. It has one. It needs one to complete its outermost shell. It only has one shell, so it can only accommodate for 2 electrons. Chlorine needs one. It has 7 already, and so what they do is they end up sharing the electrons, and that is how the bond is formed. Similarly, for water, right, 2 atoms of hydrogen bonding with 1 atom of oxygen. Oxygen has 6, it requires 2, and that gets that from sharing 1 electron each of the hydrogen, and the hydrogen shares 1 electron each of oxygen, thereby getting 2 electrons in its shell, and that is covalent bonding. Then water, right, has certain properties, primarily because there's a dipole, so it is a polar liquid, right? Uh, Molecules that have dipoles are called polar. Right? I'll just add that down over here. And the reason why they have dipoles is because even though it's covalently bonded, right, the electrons are slightly more, right, attracted towards the oxygen than the hydrogen, because the oxygen has more protons to pull the electrons towards it, and because electrons are negatively charged, oxygen develops a slight negative charge, or delta negative, and each hydrogen has a slight positive charge. And what that helps is for the hydrogen's positive to attract another water molecule's negative, and a water molecule's negative to attract a hydrogen atom's positive, right? And so these attractions, they're not formally bonds, as in they're not sticking together in that sense, but they are keeping them close together, and those are called hydrogen bonds. And they confer certain properties to water, right? That which you will not find in similar molecules, such as hydrogen sulfide. So they can dissolve ions and other polar substances, right? Water is a good transport medium for the same reason. It has anomalous behavior where at 4 degrees centigrade it achieves maximum density because it has minimum volume. And the strange thing is ice is less dense than water. It absorbs a lot of heat because it has a high specific heat capacity because of the hydrogen bonds, because it takes a lot more heat to break the hydrogen bonds and cause the water molecules to move further apart. Right, just as a recall, specific heat capacity is the amount of energy or heat required to raise temperature by 1 degree centigrade. Water molecules are cohesive, so they stick together, and adhesive, so they stick to the surface. Is very important in plants in the xylem. Right, they have high surface tension, again because of the hydrogen bonding, and so they can form a surface sort of layer on which many organisms interact and even live out their lives. That was chapter 1A1. Then coming to chapter 1 and 2, where we started talking about biological molecules. Right, so the main classes of molecules that are important in biology, there are 3 of them: carbohydrates, lipids, and proteins. We started talking about carbohydrates. And first talking about monosaccharides and disaccharides, right? So carbohydrates are a broad class of biological molecules that play an important role in living organisms. Normally we hear about them as an energy source, but that's not all they do. Organic molecules are primarily made up of carbon and hydrogen, but they also have oxygen, nitrogen, and phosphorus. And the thing about carbon is it can form 4 covalent bonds and have a tetrahedral shape. So you have a bond sticking out, a bond going into the plane of the paper, and 2 of them on the plane of the paper. And that you see like a 3D structure, like a pyramid of sorts. And so it can form straight chains or branched or unbranched chains and rings and many, right? So even though not organic, but graphite and carbon, diamond, sorry, and Buckminster Fullerstein, if you've heard of that in your IGCSE. Right, so these are all different molecules that carbon makes up, and they all have different physical properties, right? Especially comparing graphite and carbon. Graphite is soft, and diamond, so I keep saying carbon. Diamond is the hardest substance known to exist, right? And they're both made of carbon, so right, very different properties. Now, right, as for carbohydrates, right, a single molecule, the term used for it is a monomer. If there are 2 molecules, a dimer, and many of them a polymer, right? And so these are general names, but as you can see over here, if you've got the specific name of the type of molecules, so saccharide is what's used for carbohydrates, so mono. Instead of monomer, you're going monosaccharide. Instead of dimer, you're going disaccharide. And instead of polymer, we'll come to that, but you will have polysaccharides. And polymers are like huge molecules. They're called macromolecules because they're massive, right? And that is a review of what we had done yesterday. Now, coming to today. Okay. So when talking about carbohydrates, just get the pen. When talking about carbohydrates, right, some of the popular ones that we are familiar with are glucose. Then you've got sucrose, and sucrose is the regular white sugar that we use every day. And you've got starch. So that's the carbohydrate present in rice and potatoes, right? And right, lots and lots of other sources. And not only are these, right, the ready sources, of energy, but also, right, are storage form. Right? So starch is actually a storage form of carbohydrates in rice or potatoes, right, or other plants, not in animals, right? Storage form in animals is what's called glycogen, and we'll get to that. Right, but because there's so many more plants and they're part of the diet, right? So as far as an energy source is concerned, they are ready sources of energy, and they're also storage form of energy when required. Now, because they're organic molecules, right, carbohydrates are primarily, or in fact, only made up of carbon, hydrogen, and oxygen. Right, so you don't have any nitrogen or phosphorus in carbohydrates. And as you just mentioned, right, we can have monosaccharides, disaccharides, and polysaccharides. So let's start with the simple sugars, the monosaccharides. Right now, there's not just one type, right? Simple sugars basically mean that they are monomers. So they're single molecules of sugars. But they're different types depending on the number of carbon molecules, sorry, carbon atoms that make up the sugar. The general formula for monosaccharides is CnH2nOn. Right, so however many number of carbon atoms you have in the molecule, you're going to have the same number of hydrogen, the double hydrogen and the same number of oxygen. You can condense that and write it as CH2O, right? And you're taking N as a common factor out. It's the same thing. And so what we'll be talking about, right, are 3 carbon that are called triose, 5 carbon which are called pentose, and 6 carbon which are called hexose. So as the name is, triose sugars have 3 carbon atoms. And so their formula would be C3H6O3. Now, they haven't named any particular one over here, but we have come across, and they do mention it, right? That's something that will come across in A2 biology when we study about respiration, and that recording is available because we already covered that in A2 biology. So if you want to go and check it out. And so triose sugars are something that are involved in the process of respiration, like glycolysis. Just to hint that, that's all we mentioned. Then we have the pentose, which, right, are 5 carbons, and right, so you've got C5H10O5. And the ones of interest for us are ribose and deoxyribose. And these are both part of, right, the genetic material inside living organisms. So ribose is part of RNA and deoxyribose is part of DNA, right? So it helps form, right, the genetic material. It is part of their more macro structures, right? Because you've got a nucleotide base and phosphate as well. So it's not just the sugar, but the sugar is important. Right, and so the molecules are actually based on their components, right? So ribose, the ribose sugar makes up the RNA, and its full form RNA is ribo, coming from the ribo nucleic acids, right? And DNA is deoxyribo, coming from the deoxyribose nucleic acid. So when we talk about genetic material later on, we will obviously come across these. And then we've got the hexose, right, which are 6 carbons. And so their formula would be C6H12O6. Now keep in mind, this is the general formula, right? And so there are number, like, so for pentose, we've seen that there's ribose and deoxyribose. We haven't mentioned triose, but there's more than one. And there's more than one hexose as well. So the general formula doesn't really tell you anything about the structure, and that's where they actually differ, right? So even though they've got 6 carbons and 12 hydrogens and 6 oxygen atoms in the molecule, the structure of the atom is different, and that's what gives rise to the different hexose sugars, or the pentose sugars, or the triose, obviously. Now the hexose sugars are the best known monosaccharides. Having glucose, galactose, and fructose, right? So these three are the monosaccharides. Now, right, what is their three-dimensional structure, or how do they appear, right? The shape. Drawing that. So for a ribose, right, that's a pentose sugar. This is what it looks like. You have an oxygen atom, and then we got a bit lower oxygen. You've got carbons here, and you've got carbon up here. And you join all of that. That's sort of like a pentagon, right? And the rest of these are taken up by either hydrogen or OH groups. There's a hydrogen here. And that is what ribose looks like in specific, right? So deoxyribose, as the name suggests, it is lacking an oxygen at one of the carbons, right? So instead of an OH, there is just 2 hydrogen atoms attached, right? We'll see its structure when we get there. For, right, a hexose, we have glucose. And glucose has 6 carbons. So what we start off with is it looks like a hexagon. You have an oxygen and, right, carbon here, 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 right, here, and then a carbon up here. And you join those together, and so you've got your 6 carbons, and you fill in, not randomly, of course, the position matters. So you fill in the hydrogens and the OH groups. This is CH2OH, and the hydrogens and oxygen. So you've got a hydrogen coming here, and H and OH. And that is what a glucose molecule looks like. Does it look intimidating?
Yeah.
Okay, so it's probably the first time you've seen its structure.
Yeah, I guess.
Okay. Well, it's not so bad, right? First times, I mean, first impression is the last impression. I don't believe that, right? So obviously, it is not the regular kind of molecule that you come across, uh, right? We'll see this a number of times, and the best way, right? So it's obviously given in your book. The best way to become comfortable with this is to draw it. So on your own time, draw it out, right? Draw it as many times so that you memorize it, because the positions of the hydrogens and the OH groups, right, the hydroxyl groups, is important. You can't just place them anywhere. Okay.
Okay.
Now, to make it easy, right, the carbon atoms are numbered, right, and they're numbered in going clockwise, starting from oxygen, right? So the oxygen is obviously not carbon. And so, all right, you start from the oxygen and you start going clockwise and you start numbering them. So this is carbon 1, 2, 3, 4, 5, and 6. You would expect the ring to be made up of all of the 6 carbons, but no, there's one carbon hanging out right up top, and the ring is actually completed by bonding with an oxygen. And you just make sure that you have all 4 bonds on the carbons as well, right? So the bonding is complete. So numbering them, and this is like a standard method, right? It's not something that I've come up with. So you start from the oxygen, you go clockwise, you start numbering them. There's 6 carbons, obviously. That's how you go about doing that. And to make it simpler, right? What you do is that you skip the hydrogens and the carbons entirely. Right, so you've got your oxygen over here. What you do is simply, oh, that's 1 carbon, that's the 2nd carbon, 3rd carbon, 4th carbon, 5th carbon, coming back to oxygen, oh, that's the 6th carbon. Right? Does that look simpler?
Yeah.
And then you fill in the OH groups. Right? So, right, you've got an OH here and here. This one's going up. This one's coming back down again. And right, CH2OH. And you number them 1, 2, 3, 4, 5, and 6. Is that simpler?
Yeah, kind of.
Yeah. So in the book, they've actually sort of written out the hydrogens as well, but technically you can leave that out. So because right in organic chemistry, any bond that is left out on a carbon is assumed to be taken up by a hydrogen. If it's not a hydrogen that's bonding to carbon, then you specify. That's why the OH groups are written, right? The hydroxyl groups are written, and the CH2OH as a chunk is written for carbon number 6 attached to carbon number 5. But if you just count, oh, well, this carbon number 1 has only 3 bonds shown. Well, yes, the 4th one is taken up by hydrogen. It's sort of understood. Okay. So it's not that the bonding is incomplete and so it's not a stable molecule. It's a pretty stable molecule. But the book has gone the length of saying, oh well, let me change the color over there. Right, this blue. So they've actually gone the length of saying, oh well, there's hydrogens here. Okay. Right, and again, to get comfortable with this, just look at the structure, study it, draw it while looking at it, and then try to learn it by repeating it over and over again. And this is something that you will have to do multiple times, right? And for multiple processes, right? So you've got certain processes. Well, we have covered them in A2. So, right, the link reaction of glycolysis or the Krebs cycle and all the steps that are involved, you need to know them. And the only way, you can't just memorize them by looking at them, right? You need to write out the entire cycle or the process with all the steps on paper over and over again until you don't know them by heart, right? When you know them by heart, you're good. But again, right? So always practice and check. So that the same goes over here, right? All the important molecules, all the important structures, the ones that you should know, that's how you go about doing them, right? So you can do the same over here as well, right? For the ribose, we've got the oxygen, and there's one carbon, another, another, and oh look, we've got a ribose, and there's an OH, and OH, and this guy is CH2OH. And you number it, so 1, 2, 3, 4. Right, I've done it a bit too squarish. Right, it should actually be something like that. That will be better. Right, do that. Yeah, I'll do it myself. So again, the oxygen. There you go. CH2OH. We've got OH here and here and here. Right, and the numbering. Okay, makes life simple. Now, shall we continue? Any questions?
No, sir.
Okay. Now, not only are there at least 3 different types of monosaccharides, right? The popular ones, so to speak, or the more relevant ones, as far as our syllabus is concerned. There's a thing about glucose, right? That is also important for us to know biologically, and that is glucose has 2 isomers. Right, so organic molecules are one way, right? They are defined by the number of carbons they have and what are called the functional groups. So recalling organic chemistry from your IGCSE or O level, right? The functional groups, and if they have a particular shape or structure of the molecule, and obviously that is important as well, right? And so what isomers are, right? The number of carbons is not going to change. So the position of either the functional group basically, or maybe slightly the shape or structure will change, right? And that gives rise to an alternate form of the molecule. And so glucose has 2 of them. And how does it have 2 of them? So let's draw glucose again twice because there are 2 isomers. That's one of them. And then the other. Okay. So what's different about these guys? CH2OH, CH2OH. Now to avoid, remember I mentioned, right, we just draw the OH groups and the positions of the OH groups are important. Well, what's important in the isomer is the position of the OH group on carbon number 1. And so you can imagine that there are only 2. So, right, the position, this is a functional group, right? And functional groups are the one that give specific properties to the molecule and also take part in reactions. And so, right, this OH group can be below the carbon atom or above the carbon atom on carbon number 1. And so to highlight that, right over here, we've got an OH below the carbon atom. Over here, we've got it above the carbon atom. Right, and the numbering 1, 2, 3, 4, 5, and 6. Similarly, 1, 2, 3, 4, 5, and 6. So the position of the OH group on the carbon number 1 is what will give you the isomer. So the one where the OH group is below is called alpha glucose. And the one where it's above is called beta glucose. And so to remember the position of the OH group and which isomer it is, right? It's sort of, I mean, you can remember it some other way if that's what you like, but right, what I use is, oh well, this is alpha, right, which means above, and this guy is below, so like the opposite. And beta is with the B, right? So that's a, and this guy is above, so the opposite. I'm writing below because, right, it's spelled with a B, and so is beta, right? And so this guy is above, and that's how I try to remember it. Obviously, if it works for you, that's good. If not, right, you can think of it another way. What you can also do, if I redraw these on the next page, right, not to confuse you, to give you an alternate to remembering which isomer is which. Drawing that out. Pretty messy. Okay, much better. Again, we're just talking about carbon number 1 in this case. And sort of focusing on the OH, we can talk about the hydrogen. Right, so obviously if the OH is below, then the hydrogen will be on the opposite end. And so this is alpha glucose, this is beta glucose, and so write alpha for above, and the hydrogen is above, beta for below, and the hydrogen is below. Whichever one you find easier, or if there's a third way of remembering which isomer is which, right, then use that. And because, right, the positions of the hydroxyl groups are important, right, so you need to plug those in as well. Just a minute. And so you have the hydroxyl groups on carbon 2 coming down. This one's going up. This one's coming down as well. right? And CH2OH. And so the positions of all the others don't change, right? So that's the same. Nothing to worry about over there. It is only carbon number 1 where the OH is flipping, right, between alpha and beta glucose. Are you following along? Are you a bit more comfortable with this as compared to 5 minutes ago?
Yeah.
Okay. Right, so you keep seeing this again, you keep talking about it, you keep making it, right, and you will get more comfortable with it. So to the fact that you'd be sitting in class and you'll be doodling this, right? I used to doodle the structure of hexosugars or the DNA molecule or something, some others that I had learned back in the day, and so that's what I used to doodle back then. Right? Okay. Do we have time? Not enough to jump into the next part. I was hoping on continuing. Okay. So, right, to wrap this up, the isomers, the position of the OH group is what will play a role when we move on and talk about disaccharides and polysaccharides, right? So which one are bonding, right, as an effect, and you can kind of understand that, right, whether OH is right below or above, and so that would affect the shape of the molecule you end up getting. Okay, but we'll talk about it when we get there. That was just a prelude of sorts. Any questions?
No, sir.
Is it mostly clear?
Yeah.
Okay. All right, so you can review this later on. Again, it is obviously all there in the book, and we'll continue in our
Yeah.
next AS class. I'm sure.
Yeah.